my talk, it's about the nanofabrication. That is a topic a bit strange with respect to all the other topics of the seminars of PRP. But I will show you that is an essential tool for the modern research also in the bioscience field. When we're talking about nanofabrication, generally we are thinking okay. And that. So when we are talking about nanofabrication, generally we are thinking on the microelectronic industry. No? In fact, all the processes that are used for the micro nano fabrication, or the most of the processes, are generally derivating from the process that are used or are developed for the production of the microelectronic devices. But I will show you that the possibilities of the micro nanofabrication can be extended not just on this particular field, but on many other fields, in particular on the bioscience. But what is the micro nanofabrication? So the micro nanofabrication, talk about micro nanofabrication as a set of techniques and processes that are used in order to generate micro and nano structures or devices in the size range of 100 nanometer up to 1 millimeter, in the case of micro fabrication. Below the 100 nanometer in the case of the nano fabrication. But how we can realize this kind of devices? Simply by combining different techniques, in particular pattern deposition, etching techniques, and selecting them depending on the many parameters on the final device that we are creating or generating. In particular, the size of the sample, the aspect ratio that we want to achieve, the resolution, the cost, the number of sample or device that we are producing. That is extremely important, in particular in the case of research application, and the kind of material that we are using or we want to pattern, and for sure the final application, because depending on the application we are selecting different processes, different material, and different techniques. But okay. So just let me explain a bit better about micro nanofabrication, because I think most of you are not really familiar on this kind of technology. So when we are talking about micro nanofabrication, we can distinguish 4 different classes of techniques or processes. The first is for sure the patterning processes, that are the techniques that are used for the generation of micro and nanostructures, or the desired structure that I want to create. The second 2 classes of processes are the so-called pattern transfer processes, that we can distinguish between additive techniques, where we are somehow transferring the pattern that we are created by the patterning techniques in the substrate by adding some material, such as by using deposition techniques, like in the case of metal evaporator or sputtering, or growing in a specific position the material, like in the case, for example, of the electroplating or the electrochemical growth. On the contrary, on the subtractive techniques, what is happening is that I'm transferring somehow the pattern that I created by removing part of selectivity, part of the material of my substrate. In this case, we are talking about etching techniques. Finally, there is that class of metrologic techniques that are the techniques that are used in order to characterize the samples that we are producing by micro nanofabrication. That it's really important to characterize step by step what we are doing during the processes of the micro nanofabrication. In this class of techniques are included, for example, the classical optical microscopy techniques, electron microscopy techniques, in particular the SEM, and many other techniques, like, for example, the profilometer that is used in order to characterize, for example, the thickness of the thin film that I'm depositing during the additive processes, or the depth of the structure that I'm producing through the use of the etching. So this class seems to be the less important class, but consider that in the electronic industry, the instruments and the techniques used for the metrology are the largest part of the investment that companies are doing, because it's extremely important to perform this routine characterization during the fabrication. All right, so let's move about more in detail about the micro nanofabrication. So we can distinguish two different approaches for the micro and nano patterning. One is the so-called bottom-up techniques, where atomic, molecular, or microscale self-assembly are exploited in order to generate regular or defined structures, okay, with particular properties and size, features, and so on. A different approach is the so-called top-down approach, that is what is generally used for the in the lithographic approach. That is, again, the combination of these techniques in order to generate the pattern and the final structures on my sample. About the bottom-up techniques, I just give you an example. That is the so-called block of polymers lithography or patterning, where we are exploiting the properties of the block of polymers or particular kind of block of polymers that can arrange in ordered or quasi-ordered structures, and controlling the parameters during the deposition process of the block of polymers. We can, like, for example, the temperatures of the annealing, the thickness of the film that I'm depositing, the kind of block of polymers that I'm using. I can somehow control the final structures that I can achieve. For example, moving from random structures to produce cylinders or vertical hexagonal cylinders up to lines, okay? As you can imagine, this kind of approach is really convenient in terms of cost, because, for example, in the case of block of polymers, I'm just depositing a thin film of block of polymers on my sample, and by a simple treatment of the sample, I can generate this kind of structures. Unfortunately, these kind of processes are affected by the low control of the pattern that I can generate. So I'm limited in the selection of the structure that I can generate. And moreover, they are also affected by the defects. I cannot fully control the pattern that I'm generating. So this is why generally the top-down approach is what is used for the micro and nano fabrication. But what is in particular this kind of technology, such as the patterning of by bottom-up approach? So we can distinguish two different methods. The one is the so-called pattern replica, such as I'm just replicating a pattern that I have on my mask mold in the final sample. The second approach is the scanning probe patterning, where I'm using a probe that could be, like in this case, again, a light, like the use of a laser, in order to expose a suitable material in certain region in order to define the final structure that I'm generating. So starting from the beginning, so I'm depositing a suitable material that is called resist, that is generally material that is sensitive to a particular radiation or a particular probe. This resist is sensitive, v, for example, in the case of the most common technique, such as the UV lithography, to the UV light. and exposing the resist or the UV light through the mask, I can expose selectivity some region of my sample. Depending if the resist is a positive or is a negative, I can generate the final pattern on my on the resist that I deposit on my sample. In the case of the scanning probe, again, I'm just scanning my probe on top of the surface in order to define where are the regions that are exposed. And again, after dipping the sample in a suitable developer, such as a solution that is removing selectivity in my material, I can replicate or generate the pattern on my sample. There are many different kind of techniques. I just show some of them. For example, again, the most diffused and simplest is the photolithography that make use of a collimated UV light, exposing the sample through the suitable UV mask, that is a slab of glass containing the structures of generally chromium that are not transparent to UV light. There is the interference lithography that make use again of UV, in this case laser, that is interfere in order to generate some particular array or gratings. There is the so-called nanoimprinting or UV nanoimprinting that is replicating the structure present in a mold in my sample by using the heating of the sample or by using again the UV in order to cross-link my sample. Then there is the soft lithography that I will show you a bit later. That is extremely important in the case of biological application. But there is also many other techniques like microcontact printing, where I'm transferring somehow some molecules in another sample in order to define, in this case, as a chemical pattern on my sample. Or there are some more exotic techniques, like in the case of shadow mask lithography, where I'm using a stencil mask in order to deposit a suitable material, a suitable thin film in certain region. So again, like in the photolithography, I'm not using a UV light. I'm really depositing a metal that is passing through a stencil mask and is deposited just in the exposed region. In the case of scanning probe techniques, I just want to remind, for example, the optical scanning probe, like in the laser writer, that is make use of a laser that is scanned on top of the surface and is exposing resist, optical resist, or the laser drilling, where I'm not exposing a resist, but I'm removing by engraving the material by exposing to the laser. Another option is the use of particles beam, like in the case of the electron beam lithography or the ion beam lithography. In this case, I'm using a focalized electron beam or ion beam in order to expose, in the case of the electron beam, a suitable resist, or in the case of the ion beam, I am removing directly the material from the sample. There are other techniques that make use, for example, of particular kind of AFM or STM in order to generate a particular pattern, but that are quite limited techniques that are not really diffused. Okay, as I told you before, so with micro and nano fabrication or matching, we can apply these techniques for generation of samples in many different kind of field, starting from the MEMS, microfluidics, that Sonia was talking before, surface engineering, so we can use for the design of particular optics. In the case of detectors, we are working a lot with the synchrotron in order to develop a new kind of detector. And for sure, for the application in the to generate bio nanostructures. I give you some example of what we are doing in this particular field. We are working in all of this field, but in particular, we have huge activity in the application in the biology, in particular in bio research device. The first example is concerning the study of the migrating cancer cells in order to develop a new system for the investigation of the properties of the migration of the cancer cells. So, as you know, so better than me for sure, the cells are experiencing or exerting some forces, and this is how they are somehow interact with the environment where they are living. But how to measure these forces is the common technique to evaluate this, the exerted forces by the cells is the so-called traction force microscopy, consisting in the seeding of the cell on a particular hydrogel containing some beads inside. By evaluating the displacement of these beads inside the hydrogel, I can somehow evaluate also the forces that the cells are exerting to the hydrogel. As you can imagine, these techniques has some limitations. In particular, it's a bit complex and it's affected by also complex analysis and required the advanced microscopy to measure. This is why in the last year, so we exploited the use of the MEMS or NANS. The MEMS or NANS are technology that is similar to the technology that are used for the EFM, that make use of microcantilevers in order to evaluate the forces that are applied to this cantilever. With the use of the microcantilevers of the MEMS, we can obtain extremely high sensitivity, high mass resolution, very high multiplexity, and fast analysis. That are just some example of possible application. Generally, this kind of technology is performed in vacuum in order to push the sensitivity or of our device. But if I want to apply the same technology for biological application, such as, for example, to measure the force that are exerted by the cells, as you can imagine, I have to apply or introduce this kind of device in a liquid medium. Otherwise, the cells are not alive. So there are some papers in literature that are showing the possibilities to perform this kind of experiments. Unfortunately, by inserting these cantilevers in a liquid medium, there are some limitations. In particular, we have the reduction of the quality factor that I can measure with the cantilevers. And moreover, we have a problem due to the scattering of the laser. So in this kind of technology, so we are generally using a laser that is focalized on top of the cantilever. And by measuring the deflection of the laser, we can evaluate somehow the deflection of the cantilever. But in presence of a liquid medium, we have the scattering. And moreover, in presence of the cells, we have also induced scattering of the light. Also because by completely dipping the cantilever inside the liquid, when I'm seeding the cells are growing on both sides, okay? A different approach in order to solve all these limitations is the use of a microfluidic channel inserted directly in the cantilever, like it's shown in this picture here. So as you can see, you have the cantilevers and the cantilever, and it's integrated this micro channel in the cantilever that is completely sealed on the cantilever. This is a very smart approach. I mean, on the point of view of the nanofabrication is quite complex, but can provide extremely good resolution. But in the case of the application, that is extremely useful in the case of the measure or quantification of small molecules, proteins, DNA. But in the case of application of cells, as you can imagine, the size of the channels is the limited factor. So I cannot use for the most of the cells that can be used. This is why another strategy that we are developed in our lab is the use of the so-called half wetting condition, such as to have the cantilever that is wetted but just on one side. In this way, we can have avoid the limit of the scattering of the laser, because the laser side is in air, so it's not affected by the presence of the liquid. And moreover, we have also the growth of the cell on the wetted side of the cells. This is just to show how to produce this kind of device. Simply, as I showed before, there is a first step, that is the patterning step, by using the UV optical lithography in order to define the suitable pattern. And then with a double step etching process. The first is a dry etching process, such as the removal to the use of the plasma of the silicon nitride, and finally with the wet etching of the substrate in order to open completely the silicon and generate the suspended cantilever. I can fabricate this cantilever that you can see. It's simply cantilever that is inserted in a membrane, silicon nitride membrane. We evaluated the difference of this kind of cantilevers in 3 different conditions, such as in the fully wet condition, in half wet condition, and in air. Obviously, in air, the Q factor is much higher than in half wet and full wet, but you can see that in half wet conditions, we have an improvement of the Q factor of 5 times more than in fully wet conditions. And this way, we can achieve a quite good sensitivity in terms of the force of the cells on the that are present on the cantilever. We tested this technology by using 2 different cell lines, the MDA and the MSF cells. The first is an highly invasive cells, while the second one is a low invasive. And we seeded the cell on top of our cantilevers, and we inserted the device to the use of suitable liquid cells inside the AFM instrument. As you can see here, you have 2 SEM images of the cells present on top of the cantilevers. And we demonstrated that in half wet condition, the blue line, with respect to fully wet conditions, we have a reduction of the noise of the signal that we are collecting. And moreover, we can increase the sensitivity of our system. Starting from this study, we observed also that the cells are somehow prefer to stay on the gap of the cantilever, as you can see here. No, this is, for instance, image of the cells. Starting from this observation, we decided to develop a new kind of device or substrate in order to characterize the mobility of these cells in presence of different structures on the sample. We produced the sample at the same way of the before, with optical microscopy and dry and wet etching. And we observed that increasing the size of the gap for MDA cells, for small gaps, such as a gap of 2 microns, we observe that the cells prefer to stay on the groove that we created. And moreover, they assume a shape that is elongated along the gap present on the sample. This phenomenon is decreasing by increasing the size of the gap. The MCF cells instead seems to be less responsive with the presence of these open structures on the sample. That are some images, fluorescence images of the same cells, and you can see the nucleus of the cells are perfectly aligned with the micro gap of the sample. We evaluated also the cell orientation with respect to the gap, and it's confirmed that the MDA cells are perfectly aligned or pretty perfectly aligned with the gap. Instead, the MCF are not presenting the same behavior than MDA, so you have a much random distribution of the orientation of the cells. And here we also evaluated the migration of the cells in presence of the gap, and you can see that the cells prefer to move on the gap, and when they reach the gap, in particular for the small one, they prefer to stay there, and they are moving along the gap. And this is again effect. This effect is less evident in the case of larger gaps, like in the 4 microns, and is again less evident for 8 microns. And here we just track the velocity of the lines to reach the gap. So just to say that somehow the pattern of the sample is somehow affecting the properties or the behavior of the cells that are seated on top of this. Moreover, these kind of structures here can be exploited also in order to somehow drive the cells in particular positions. And now we are evaluating also the opportunity to use similar devices for the use in the cryo-EM, because in this way we have completely open structures. We can somehow increasing the quality of the imaging that we can collect from this kind of the cells. And moreover, we are somehow driving again the cells in particular position that makes easier and faster the sample preparation for the cryo-EM. Another application that is strongly related with the previous one is the use of these nanopillars or micropillars, again for the use as a sensor. In this case, it's for the detection of biomarkers in a solution. In this case, we are not using the planar geometry, but we rotated the geometry in order to have these cantilever that are vertical with respect to surface. Moreover, by generating an array of pillars, we can somehow exploiting the suprahydrophobic properties of this kind of devices in order to have a kind of partial wetting conditions. In this way, just the top of the pillar is the sensitive part and is the part that is wetted by the solution containing the biomarkers, while the rest of the pillar is in air and can vibrate it easily because it is not completely immersed in the solution. Again, so we can get a higher quality factor. In this case, we didn't use the UV lithography because of the limit of the final resolution that you can get with the UV lithography, but we used the electron beam lithography. The electron beam lithography is quite simple technique that makes use of a beam, the electron beam that is focused on the substrate, like it's similar to the SEM. So you have a beam that is focalized on top of the surface. In the case of the SEM, so you are scanning all over the sample in order to collect the image of the sample. In the case of the electron beam, the beam is scanned just in some region of the sample, and just that region you are inducing the change in the solubility of the resins that is present on the surface. After dipping the sample again in the developer, you can remove that part that is exposed. With the electron beam lithography, we can reach much higher resolution with respect to the optical lithography. On the contrary, we are a bit limited in the number of samples that you can create and in the write speed. Such as to expose the same area that you can expose with optical lithography, you need order of magnitude more time. Okay, so this is why when you are thinking of your experiment, you have to design and select properly the techniques or the tool that you are using in order to produce the sample. After the exposure, we produce the pillars by using again a dry etching process. In this case, we use a process in ICP RIE instrument, in particular by exploiting the so-called Bosch process. In the Bosch process, we have a cycle of removal and passivation of the walls, like is described in this picture. I don't want to enter too much in detail, but it's just to tell that with this technology, in this way, you can reach extremely high aspect ratio in the final structure that you can get. This aspect ratio cannot achieve, for example, with wet processes. As you can see here, the top of the pillar is on the range of 1-2 microns, while the height of this pillar is on the range of 10-15 microns. So more than 1 to 10 aspect ratio, final aspect ratio. Moreover, by controlling precisely the etching process, you can control also the shape of this pillar. As you can see, the pillar is a bit tapered, and in this way you can control the size of the bottom of the pillar. In this way, you can control also the frequency of the vibration of these pillars. We tested the similar architecture, and we tested different readout method. The first, that is the classical laser-based optical readout, that is exploiting it's called the modulation of the laser depending on the vibration of the pillar. While that is extremely sensitive method, while with CCD, parallel optical readout, we are just measuring or highlighting the vibration of the laser simply by collecting the image with the CCD. In this way, with the laser, we can monitor just one single pillar each time. With this method, we can evaluate the resonance frequency of tens of pillars simultaneously. Another lithography technique is the nano-imprinting lithography, that is also extremely important in the case of bioscience application. It's a really simple techniques that make use of a mold containing some feature that I'm replicating on the substrate, like in the case of the preparation of the waffle. So I have the structures on the plate here, the heated plate here, and I'm just pressing the plate on the waffle, so I can cook and obtain the same pattern. Okay. In the case of thermal nano-imprinting lithography, we are using a mold containing the structures, and by heating the sample on top of which I deposited a thin film of thermoplastic material, like a polymer, and pressing the mold on the resist, I can simply replicate the structures that are present on the mold on the resist. Okay. After the removal of the residual layer that I have at the bottom of the structure here, I can generate the replica of the pattern on my sample. In the case of UV curable nanoimprinted lithography, we are using not thermoplastic material, but a resin that is sensitive to the UV light. Again, we are pressing the mold on the sample with the resin on top, and after the exposure of the exposure of the sample to the UV light, I'm inducing the cross-linking of this resin, and I can achieve the final structure, the replica of the structures on my sample. The key advantage of the nanoimprinting is for sure the high resolution that you can achieve. So we are replicating the structure that you have on the mold. If it takes a lot of time in order to generate the mold, then it's quite easy to replicate the structure on the mold on the sample. And you can replicate as many times as you want on the sample. That is a quite convenient technique in order to generate high resolution structures in a large number of samples. That is quite important, for example, in the application of a biological sample, then when it is required to have a large number of samples in order to have have a possibility to perform many different processes. By using the nanoimprinted lithography, for example, we developed a new kind of NEA sensor, such as a nanoelectrode array sensor, that make use of the thin film of polycarbonate, where are physiosorbed some proteins, and by the use of cyclic voltammetry, it was possible to measure the amount of protein present on top of our sample. In this way, we generate a lot of samples in a pretty easy way. And you can easily control also the feature of the final sample that you have. Finally, I want to talk a bit about the soft lithography. Soft lithography is the technique that has been developed mostly for the application in biological application. Soft means that these techniques are not making use of energetic particles like electrons and ions or radiations, like in the case of the UV photolithography. But instead, it makes use of soft elastomeric stamp or mold for the replica of the sample. The first person that researcher that developed this kind of technology is Professor Whiteside. And why it's extremely important in the case of biological application is the importance is because of the low cost of these techniques, because it makes use of very simple tools in order to generate the micro nanostructures, and for the final achievable structure that you can obtain with this. The most diffused and used material that is used for this kind of techniques is the PDMS, that is a silicon elastomer. It's very simple and is quite cheap material. That is extremely biocompatible, both with cells, foods, and small molecules. It's chemically inert, and it's very easy to be processed. With PDMS, you can reach, okay, the best resolution is written here, 210 nanometers, but no, I never achieved this kind of resolution. But you can, with PDMS, PDMS is very simple. So you have two precursor. One is the elastomer and one is the catalyst. By mixing both of them and heating them, you can co-regenerate the elastomer. By changing the ratio between elastomer and catalyst, you can also regulate somehow the stiffness, the final stiffness of the elastomer. And this is why also it's very diffused. How to use this PDMS? Simply, you are generating a mold that could be easily a substrate where I perform the UV lithography, and then by casting the PDMS on top of this and treating the material with a thermal treatment, I can induce the cross-link of PDMS, and the final step is just a removal of the peel-off of the PDMS from the mold. Again, I can replicate as many times as I want the structure that I have on the mold. This technique, for example, in our lab has been used in order to generate nanopattern and substrate for that have been used as a precursor for the differentiation of the cells, like in the case of this neuronal precursor. And okay, this is simply the process that we used for the pattern generation. And here you have some example of the neurons grown on the substrate. And we observed that in effect, so this kind of cells, but generally all the cells are likes to grow on the PDMS. And moreover, since you can regulate the size of the structure that you can obtain in this way, you can somehow regulate the stiffness that the cells are perceiving by growing there. So you have, in any case, a limited range of stiffness that you can obtain from PDMS. By adding these nanostructures, the cells are perceiving that these nanostructures are working like a reduced stiffness and likes to grow in this kind of substrate. Okay, that are just some example. There are many others that we also developed in our facility. Just think that's what you can imagine to produce. In most of cases, you can realize with a micro nanofabrication. Okay, so you need a bit of experience in order to select the suitable materials, suitable processes, and you have to define, okay, the number of samples that you need, but in any case, so you can do it in the most of case. And just let me conclude with a bit of propaganda of our facility. So we have a nanofabrication facility here in the Basovizza at the synchrotron. We have all the techniques that I show you for patterning, for the etching, for the deposition, and also all the tools for the metrology. And the policy of our facility is to collaborate with all the users that want, and also to promote also the user to enter in the facility and to try to produce their own samples, in particular, okay, with our support. But it's extremely important for us also that people know how to produce this kind of sample in order to also to perceive what are the limits that you can have in the fabrication and micro nano fabrication. So if you want to apply, just contact me. Here is the webpage of the website. You can find many other information about it. And we wait for you.